This screencast is going to be about a couple more inheritance patterns, multiple allele traits, and sex-linked traits. So we'll start with multiple allele, which is really just what it sounds like. It's when a trait has, instead of just two options for alleles, like big B, little b, some traits actually have three or more alleles. So there's three or more forms of the gene that could be there. And the example you usually see about this is human blood type. So human blood type, not the positive negative, that's a simple dominant recessive trait, but the other part of human blood type has three different alleles. So can you think of what they might be? So the answer to that is A, B, and O. So what we mean when we say blood type is we're concerned about blood type because of an immune response that can happen if you have the wrong type of blood in you. There are what's known as antigens on the surface of the blood cells, and we talked about this a little when we were talking about cell membrane structure, but they're basically glycoproteins that are embedded in the membrane of the blood cells, and those are recognized by the immune system as being from you or foreign, and the Rh antigen and also the A, B, and O blood groups tend to cause a large immune response if the wrong type of blood ends up in you. And you could have a out of control sort of inflammatory response and even die if you end up having the wrong blood type given to you, say in a blood transfusion. So the way the inheritance pattern for O and A and B works is that O is recessive to both A and B, and then A and B are codominant to each other. So if you have type A, you have an A antigen on your blood cells. If you have type B, you have a B antigen on your blood cells. If you have type AB, you have both the A antigen and the B antigen on your blood cells. And if you have type O, you have neither the A antigen nor the B antigen on your blood cells. So since you have two alleles for every gene, take a minute and pause this. Here's all the possible combinations of allele. What blood type would each of these people have if this was their allele combination? So AA would be type A. A and B would be type AB. OB would be type B because B is dominant over O. BA, well, that's just AB again. OO, that would be blood type O. AO would be blood type A because A is also dominant over O. And then BB would be blood type B. So we can do Punnett squares with these blood types. Here's a problem. A heterozygous person with type B blood has children with a person with type AB blood. What are the predicted phenotypic, so physical, and genotypic, genes, ratios of the offspring? So let's start by figuring out what are the genotypes of the parents, because we need to know that to do the Punnett square. So if a person is heterozygous and they have type B blood, that means they have two different alleles and their phenotype is B, so they must have B and O for their alleles. And then a person with type AB blood, well, their alleles must be A and B. So here are our two parents. And here's my Punnett square set up, AB times BO. I'll just bring my alleles down into the squares now. And so here is my result. I've got my genotype ratio is 1AB to 1BB to 1AO to 1BO, or 25% of each of those. And my phenotype ratio is 2 type B to 1 type A to 1 type AB, because these two individuals both will have type B blood. So there's a 50% chance that the baby would be type B, 25% chance each it would be AB or A. The other inheritance pattern we're going to talk about in this is sex-linked traits. And so for most traits, it doesn't matter whether you're a male or a female, Inheritance-wise, you'd have the same odds of having those traits. So like a male isn't more likely to have blue eyes than a female, for example. But there are some traits that do depend on your biological sex to determine your likelihood of having them. And those are genes that are found on the sex-determining X and Y chromosomes. So quick review, what is the genotype of a genetically male person and what is the genotype of a genetically female person? So the answer to that is a male would be XY and a female would be XX. Is it possible through non-disjunction to have some other combination of chromosomes? Yes, it is. Some of these are fatal. For example, you can't live with just Y and no X. But some of them, for example, you could have XXX or XXY, and those people would survive and probably be mostly fine, although they would have some physical differences. For the sake of our discussion today, though, we're just going to focus on these two most common options. And then just a note on this slide, all of the chromosomes that are not the sex chromosomes can also be called autosomes. So you have sex chromosomes and then you have autosomes, which are just all the rest of the chromosomes. And a trait that is inherited on the non-sex chromosomes is called autosomal. So here's a picture of an X chromosome and a Y chromosome when they're in their duplicated form. So this big one is the X and this little one is the Y. So you should notice right away the Y chromosome is tiny. It's very, very small compared to the X. And so what that means is there are some genes that you have, a copy on the Y and a matching copy on the X, but there are some genes that are only found on the X chromosome and are not found on the Y chromosome. So what that means is for those genes, a female will have 2 copies of those genes, so 2 alleles like normal, because she has 2 X chromosomes, but a male will only have 1 copy of those genes because he only has 1 X chromosome. So it turns out that males are actually kind of chromosomally deficient. So one important example of this type of trait is hemophilia. So I certain the most common kind of hemophilia, the gene that causes that is found only on the X chromosome. So there's a gene that's important for clotting blood. If you don't know what hemophilia is, it's a disease where your blood doesn't clot properly. So if you get a cut or a bruise, you might just bleed and bleed and bleed and not be able to, stop bleeding, which is obviously a problem. So if that very important blood clotting gene that's on the X chromosome is damaged in some way, you'll have a problem clotting your blood if you don't have a good copy of that gene. So let's try a practice problem. Let's say we have a mother who does not have hemophilia, but she is a carrier for the hemophilia gene, and she has a child with a father who is perfectly normal, doesn't have any hemophilia genes. What are the chances of their child having hemophilia? So normally with a recessive trait like this, you'd say, well, if the father's not a carrier, then none of their kids would have hemophilia, right? Because for a recessive trait, you have to get a gene from both parents in order to have it. However, with these sex-linked traits, it turns out that's not quite the case. So the way you're going to set up Punnett squares for sex-linked traits is you're going to start by just writing XX for the mother and XY for the father. And then to represent the fact that the gene is on the X chromosome, we're going to use little superscript letters on the X chromosome to represent the gene. So in this case, I'm using N for normal because normal is dominant. So our female has 1 X chromosome with a normal copy of the gene and 1 X chromosome with a abnormal hemophilia copy of the gene. And again, she's okay because she still has 1 gene that's making the blood clotting protein. And then our father has an X chromosome with a normal copy of the gene and a Y chromosome that just doesn't have the gene at all because this gene is not on the Y chromosome. So the Y chromosome, you're not going to write anything up here. Then we're going to do our Punnett square just like normal. So we'll bring down all of our alleles. And here are our results. So if we look at each of these boxes, the two XX individuals are females and the two XY individuals are males. So overall, as we'd expect in any cross between a man and a woman, there's a 50% chance of them having a female offspring and a 50% chance of them having a male offspring. Now let's look at the hemophilia status. So this female, X big N, is totally normal. She has 2 normal copies of the gene. She's not a carrier, and she's not hemophiliac. This daughter is a carrier, so she has 1 copy of the hemophilia gene, but she does not have hemophilia. This individual, this son, has the normal gene, so he does not have hemophilia. But this son has the hemophilia gene. And since there's no gene on the Y chromosome, he has hemophilia, because in the case of males, it only takes 1 copy of the bad gene in order for them to have that trait. All right, let's try another problem. Red-green color blindness is another sex-linked recessive gene. So if we had a father with red-green color blindness and he has children with a normal non-carrier mother, what are the chances they will have a colorblind child? So I'd like you to pause and see if you can do this problem on your own, and then you can continue and I'll show you how to do it on the next slide. So for our parent genotypes, we have our normal non-carrier mother. So I'm just using N's for normal again. So she's X big N, and then our colorblind father, X little n, Y. And again, with these kind of traits, we don't put any gene on the Y because it doesn't carry that gene. I'm gonna fill out our table. And we get this. So going through these, we see that these are the females and these are the males. So we have 50% daughter who is a carrier, but she's not colorblind. And then we have 50% a son who has totally normal vision. So in this particular cross, none of their children will be colorblind. So here's another question for you to sort of pause and consider on your own. We know that these sex-linked recessive disorders, such as hemophilia, are much more common in males than females. So why is that? What it really comes down to is that in order for a female to have a sex-linked recessive disorder, she would have to have 2 copies of the affected gene. She would have to get 1 copy from her dad and 1 copy from her mom in order to actually have the disorder. The male, however, only has 1 copy of the gene. So for the male, it only takes 1 copy of that gene for him to have the disorder. He only has to get it from his mom. So essentially, the female has to have bad luck and get 2 bad alleles, where the male only needs to have 1 in order to have the disorder. So for that reason, it's much more likely for a male to be affected by the condition than a female. So the last thing we're going to look at in this section is an example of a sex-linked trait that is also codominant, because we can have traits that fall into multiple categories. So did you know that calico and tortoiseshell cats, so cats that have both black and the orange or yellow color on them, that are both black and orange, are all female, except for the very, very rare XXY male, so a male that has a chromosomal non-disjunction. Almost all of them are females. I have a female calico cat myself. So why is that? It's because the gene for orange fur and the gene for black fur are codominant alleles and they are sex-linked, so they are found on the X chromosome. So in order for a cat to have both orange and black fur, it has to have one X chromosome with an orange allele and one X chromosome with a black allele. And only females in most cases have two X chromosomes, so only females can have those two colors. So if we look at all of our possible combinations, and the gene for having sort of the white chest, the tuxedo gene, that's a whole different gene. But just looking at the black and orange part, if we look at all our options, we could have XBXB, which would be a black female, XOXO, which would be an orange female, XBY would be a black male, XOY would be an orange male, and then XBXO would be a calico or tortoiseshell female cat. So sometimes in media, you'll see calico cats depicted as males. I think there's one in Big Hero 6, for example. So you can now, look at those and say, like, that's wrong, because that cat should be a female.